This week, we're going to talk about nutrition and food crop biotechnology. I want to start by posing a question to you. What innovations do you think have saved the most human lives? Do you think it's something like seatbelts or chemotherapeutic drugs that help treat cancer? What invention have humans come up with that have saved the most lives? Take a second to think about it. Why is that the invention that you've chosen? How many lives do you think it saved? Now keep that in the back of your head because the answer to this question might surprise you. It's actually synthetic fertilizers. These synthetic fertilizers actually don't help people directly at all. They're applied to plants and they help plants become more productive. They help plants produce more food for people. The invention of synthetic fertilizers helped increase the global food production so significantly that it's estimated that about 2.7 billion people who might otherwise have starved were able to thrive because of synthetic fertilizers. I think that's kind of an astounding number and really exciting to see something related to food and crop health at the top of this list. Right after synthetic fertilizers are blood groups. So you might know what blood type you are. Maybe you're A positive, maybe you're B negative or O negative. Regardless of what blood type you are, the identification of what blood type you are helps doctors figure out what type of blood you can receive if you need a blood transfusion, and who might be good matches for things like organs should you need a transplant. So before we understood blood groups, things like blood transfusions were much less likely to succeed. And so the identification of blood groups also has saved over 1 billion human lives. Now, just after blood groups is smallpox vaccine, and then right after that is actually wheat, a special type of wheat, which we will also talk about in this lecture, kind of went hand in hand with synthetic fertilizers in increasing the amount of food that we produce, and therefore saving lots of human lives by making sure that people had enough nutrition. Now, a nutritionally balanced diet is important for a healthy life. You need enough protein, vitamins, fats, minerals, and carbohydrates in order for your body to have fuel and all of the building blocks it needs to keep itself running day to day. If you consume too much of one thing and not enough of another, you can put your metabolism and put your body sort of out of whack, and that can lead to health problems. So it's important to make sure that your diet is composed of enough of the things that you need in order for your body to be working correctly. Now, one really important part of your body's functioning correctly is protein. The protein that we eat is actually digested into amino acids and then remade into new proteins. What do I mean by that? Well, if you were to eat beef or chicken or some other source of protein, maybe soybeans, your digestive system will actually take the proteins that are found in cow muscle or soybean seeds, and it will break it down into its individual parts, into amino acids. And then the cells in your body will actually recycle those amino acids into making new protein. And if you don't have enough amino acids, then you can't make all the proteins that your cells need in order to function correctly. Maybe you don't have enough amino acids to make polymerases to drive, the expression of genes in your genome. Maybe you don't make enough of ribosomes to translate RNAs into new proteins, right? It's really important to have all of these building blocks in sufficient quantity for your cells to function correctly. Now, kind of amazingly, your body can actually synthesize 11 of the 20 amino acids that are the building blocks of proteins. That means that they can actually make the molecules from scratch inside of your cells. And that's pretty cool. That means that you actually don't need to consume a lot of protein that has alanines in it, for example, because your body is capable of making them. But there are essential amino acids, and these cannot be synthesized within the body. So you actually have to eat enough protein that has these amino acids within them in order to have a sufficient source of these amino acids to get incorporated into new proteins. So here are the 9 that are essential amino acids. And these 3 at the bottom, valine, leucine, and isoleucine, These are proteins that are really essential for you to consume in high quantity. They account for 30 to 40% of the essential amino acids that you need to consume. So when we look for protein sources, we want to look for things that are high in valine, leucine, and isoleucine. So although all amino acids are required for body growth, these essential amino acids are really important to get in your diet because you can't make them and you need to have them. So each food that you eat has a unique profile of amino acids, right? Rice is obviously like a different organism than a cow. It's also different from a fish. And each of these organisms have their own kind of proteins that they require to function. And so they will have like different compositions. So you need to consume the right types of dietary proteins in order for you to get enough of the proteins you need. What do I mean by that? Okay, well, often people eat rice and beans together. And although that is nice because they taste good together, that's also pretty practical. Rice is a pretty complete protein. It has the essential, almost all of the essential amino acids that you need, but it's a little bit low in lysine. It's pretty high in methionine, which is helpful, but it doesn't have quite enough lysine to be sufficient for being the only type of food that you consume. You would eventually need to eat something else to get enough lysine. Now, beans, on the other hand, are high in lysine, but low in methionine. So the two complement each other really nicely because if you eat both of them together, then you get enough of the essential amino acids that you need to function well. Quinoa, on the other hand, is what's called a complete protein. So it has all of the amino acids or essential amino acids that you need to consume in your diet at sufficiently high quantities that you can consume just quinoa, for example, and get all of the protein that you need in order to function. Animal products are similarly complete proteins and are a good source of all of these things. Now, even in animals, some of these amino acids are consumed. Now, so a big building block, a major part of an amino acid is actually nitrogen. And so nitrogen is in your food actually comes from the air. So nitrogen is fixed from a gaseous form into ammonia, which plants can then uptake and incorporate into their seeds. So for rice, that would be into the grain or into other parts of the plant in order for the plant to be able to make protein. And that conversion of atmospheric nitrogen into a form that the plant can use to build proteins is actually converted by bacteria. These bacteria we will talk about a lot, but it's kind of interesting that they're the ones pulling nitrogen out of the atmosphere and turning it into a form that then plants use to make proteins that then animals eat. And then we eventually consume either in the form directly of animal meat or other animal products. So all of the nitrogen that's in your food is actually coming from the air, or it was at one point. Now, plants form unique relationships with bacteria in order to get this nitrogen. Some plants form symbiotic root nodules. So they actually make little homes for bacteria to live within their roots. The bacteria, when they live within these root nodules, will fix nitrogen. They'll actually take in nitrogen gas, and then they will deliver ammonia, or a form of nitrogen that the plants can use, to the plant cells. And in return, they'll get sugar from the plant. Remember, the plant is fixing carbon dioxide into sugars that can then be consumed by other organisms. So there's this really nice symbiotic relationship between plants and bacteria, where the bacteria are giving the plants nitrogen and the plants are giving the bacteria sugar. But not all plants form these root nodules. Some of them just associate with bacteria. So there will be bacteria living on the surface of the root system, and those bacteria sometimes fix nitrogen, mostly for themselves, but they will fix excess occasionally, and the plant will benefit from having those bacteria nearby because they'll be able to get nitrogen from them. There are also free-living nitrogen bacteria, which are present more in bulk soil. There's some of them flying kind of around in air. They ride air currents, and they are fixing nitrogen, again, primarily for themselves, but other organisms can take advantage of the fixed nitrogen that the bacteria are producing when, for example, the bacteria dies, other organisms can take advantage of the nitrogen that they've fixed. Now, here is what these root nodules look like. I think they're pretty amazing. All of these things that look like little beans, they're actually stuffed full of bacteria that are fixing nitrogen for the plant. The way these nodules form it's actually quite interesting. It's a very intricate relationship between the plant and the bacteria, where there are these tiny root hair structures on the surface of these roots. They're really small, and so they're difficult to see in the pictures that we have here. But a bacteria will release some chemical signals that the root then recognize, and the root will send back chemical signals to the bacteria. That will cause an infection thread to form, basically a little channel in which a bacteria can enter into the plant. And once they're inside of a deeper layer of the plant root, they'll begin to replicate and then differentiate into what's called a bacteroid. So this is a version of the bacteria that is optimal for fixing nitrogen and receiving carbon directly from the plant. Show you, I think, on the next slide what this looks like. Yeah. So these bacteroids are sometimes elongated versions of the bacteria. They tend to lose their own cell wall and permeate their cell membrane. So they sort of become one with the plant cell, but they are distinct enough that, they are still capable of fixing nitrogen and of receiving sugars from the plant. They just they enter a new cellular state. They kind of don't look as much like bacteria as they did before. But even though they do that, this video should play, there are many of the bacteria that form these root nodules are actually capable of living on their own. And so here's just a video where a researcher cut open one of these root nodules, start it again from the beginning, and then squeezed it. And you can see this sort of looks like really fine powder exuding from the root nodule. As they squeeze it, you can see it kind of puff out. That's all bacteria. And so some bacteria, even when they're in this bacteroid sort of state, retain the ability to reform their cell wall and then start to live independently again. So if you cut them open, you can actually culture some of these bacteria, which is really interesting. Okay, but this nodulation, this is a very specialized process that only occurs in some plants. It is common in legumes, and that means it's common in things like peas and peanuts and beans of different kinds. But many other plants, for example, potatoes and rice, don't form associations with nitrogen fixing bacteria and can't get a lot of nitrogen from this type of interaction. And so they're stuck sort of getting fixed nitrogen at lower quantities than the legume species and having to find other ways of getting nitrogen. So one solution that was recently discovered in maize is this mucilage. So these are actually brace roots from maize. Here's a maize plant. And it produces these brace roots, which are sort of truncated roots that are coming out above ground. And they will produce a lot of mucilage. So this kind of clear stuff that you see here, that's not water. That's something that the roots have exuded. And that mucilage will actually trap nitrogen-fixing bacteria that happen to be traveling through the air. And then it will drip them down into the soil by their root systems so that the nitrogen-fixing bacteria will then be making nitrogen in a place that the plant can access. So I think that's actually a pretty cool example of maize, which is not able to form root nodules, figuring out how to get nitrogen for itself. Now, not every variety of maize does this. In fact, most of the ones that we grow commercially don't have this capability, but it's an interesting example of a wild kind of relative of the maize we usually eat, being able to form an association with nitrogen-fixing bacteria. Okay, so here's where we get to synthetic fertilizer. In 1910, the Haber-Bosch process was invented for chemically fixing nitrogen gas. So instead of relying on bacteria to fix atmospheric nitrogen into a form that's what we call bioavailable or able to be used by plants and people to build proteins. They were fixing the nitrogen gas through a chemical process. So here's just sort of an outline of what this process looks like. You can read the Wikipedia page about the Haber-Bosch process if you're curious about these steps. Kind of like this simplified view of the Haber-Bosch process better, where you're taking nitrogen gas and hydrogen gas and you are mixing them together and applying a lot of heat in order to get ammonia, or the fixed version of nitrogen, to be made. This process is very energy intensive, but it's also very powerful because this fixed nitrogen is what we then apply to plants as chemical fertilizer in order to get them to grow. So now the plants are no longer dependent on the microbes growing in or around them in order to be able to produce proteins and, get quite productive, get quite big. They can rely on chemical fertilizers supplied by people. Now, these chemists, Fritz Haber and Carl Bosch, won Nobel Prizes in chemistry for developing the Haber-Bosch process. So the invention of this nitrogen fertilizer dramatically increases food production. The world population has grown very significantly, right? We're more than 7 billion people in the world right now, close to 8 billion, in fact. But the world population without synthetic fertilizer is predicted to have been much lower, about half the population, because we would not have been able to feed as many people without the synthetic fertilizer. So this red line shows you where we would be without this synthetic fertilizer, and the gray line is just showing the proportion of the world population that is able to be fed because we have invented these synthetic fertilizers. And they were, again, invented right here around 1910. So a really powerful invention. But like I mentioned before, it wasn't just fertilizer. There was a simultaneous invention of semi-dwarf food crops that enabled fertilizer to boost crop yields. So here's a picture on the left of semi-dwarf wheat. This wheat is shorter than conventional wheat. It has sort of a sturdier stem. The cells, individual cells within the plant, actually don't get quite as big, and so you get a more compact structure. And that's really helpful because as you apply fertilizer to these plants, they will both get taller, but the semi-dwarf plant won't get so tall that it's a problem, whereas the conventional wheat plants, or the conventional plants of even rice or barley, will get so tall that they'll lodge, and that means they'll fall over in response to kind of gusty winds, stormy conditions. And when the plants fall over like this, it becomes very difficult for them to set seed and for farmers to harvest the grain. And so lodging usually results in large losses of agricultural product. So these semi-dwarf plants were really key to farmers being able to utilize the fertilizer because they could apply fertilizer to these plants and get an increase in grain yield instead of just more straw biomass. So I like to point out the invention of this semi-dwarf wheat because I don't think it's quite as famous, though agronomist Norman Borlaug, who is the person that developed semi-dwarf wheat, actually won a Nobel Prize for his efforts as well. He won it I think 30 years after the Haber and Bosch Nobel Prize was given out. So kind of exciting. Now there are problems with applying lots of nitrogen fertilizer to farms. You can get runoff. So if you apply more fertilizer to a farm than the plants actually need, then the excess nitrogen can end up in waterways where organisms that are essentially limited in growth by the amount of nitrogen. Remember, like all nitrogen has to get fixed from this gaseous form by some sort of microbe. And there aren't a lot of those nitrogen fixing bacteria naturally in the world. So if you supply a lot of synthetic fertilizer, you can get big blooms of organisms. In this case, this is an algae growing in a lake, and the algae actually grow so vigorously that they use up a lot of oxygen in the lake, and that kills off a lot of other things, like a lot of other fish and animals that require oxygen within the water to survive. And so excess nitrogen running off of farms can be really problematic for ecosystems near farms, and even actually pretty far away from them, as these chemicals can travel pretty far. Okay, so can we move away from synthetic fertilizer? This is a question that people have been asking and trying to figure out an answer to for at least the last 10 or 15 years. In 2012, the Gates Foundation started funding efforts to genetically engineer cereal crops to produce their own nitrogen. So cereal crops are things like wheat, barley, rye, rice, maize, and these are plants that cannot form root nodules. They cannot form those symbiotic associations with nitrogen-fixing bacteria. And so they are pretty reliant on synthetic fertilizers in order to get enough nitrogen in order to produce grain at the quantities that we're used to them producing at. Now, we could just stop applying nitrogen fertilizer to these plants, but we would drastically reduce the amount of food that we're able to produce globally. And so it would be, a big problem for the human population if we did that. So another solution is just to figure out how can we modify these plants to be able to produce their own nitrogen. Now, there's been some progress towards trying to do this. It's a really challenging goal. It's really hard to get cereal crops to fix their own nitrogen or to form associations with nitrogen-fixing bacteria. So scientists have taken on different approaches towards trying to get nitrogen-fixing cereals created. This effort is to try to get barley to associate with rhizobacteria, which is a type of bacteria that would form root nodules on legumes, on bean plants. But they don't talk to each other. Remember, the first step in forming a root nodule was a chemical communication between the bacteria and the plant root cells. And the barley plants just don't talk to the rhizobacteria correctly. They actually just don't make the right chemicals in order to attract the bacteria. And so these scientists have been able to engineer plants to produce a chemical that nitrogen-fixing bacteria recognize and respond to. Here's the chemical that they're producing, and on these plates below, you can see in black and white a little barley plant that has bacteria applied to them. And the bacteria are engineered to luminesce, to produce a little bit of light when they detect this compound. So usually they would detect this compound and begin some of the kind of nodule formation steps. But just as a visual signal of kind of where they are in this process, the scientists also added the capacity for them to make light. And what you can see is that when the bacteria are put onto barley plants that have been engineered to produce this compound, they do in fact produce light. But when the bacteria are put onto wild type plants or unengineered plants, they do not. And so this is a nice demonstration that the plants have been successfully engineered to produce this compound, which is a good first step towards getting this kind of trans-kingdom signaling, the signaling between the plant and the bacteria, to happen between a plant variety that would not usually talk to this bacteria and the nitrogen fixer. So it's an exciting first step. There's obviously quite a long way to go, but I encourage you to kind of keep your eye out for stuff like this. As over the next 10 years, we hope that these types of solutions just mature and become even more kind of complex and close to the nitrogen fixation that's happening in legumes. Okay, an alternative approach is not to worry about root nodules, but just to make free-living nitrogen-fixing bacteria more productive. Remember, there are some bacteria that just live on the surface or in the bulk, on the surface of roots or in bulk soil, and those do fix some nitrogen. And maybe if you just engineer them to fix a lot more nitrogen, then you don't need to form those really specialized root nodules. You can just get enough nitrogen being made by these free-living organisms for the plant to survive. So in a pair of engineers at Stanford, this is Professor Beth Satley and her graduate student, Tim Schnabel, were able to engineer a strain of azospirillum to fix more nitrogen. And this is a free-living bacteria, but it doesn't produce a whole lot of nitrogen unless it itself needs nitrogen. And so they were able to kind of deregulate its nitrogen fixing. And they're hoping that they will have a product that is something farmers can buy that consists of a bunch of these engineered bacteria that are grown up in some sort of fermentation in a lab, and then freeze dried and delivered to farms where they are put out into fields. The bacteria would then revive, they would rehydrate, and they would start to replicate. And so they'd make copies of themselves and then start to fix nitrogen for the plant. So the company that Tim started is called Switch Bioworks, and this is their goal. It's a really exciting goal to see nitrogen fertilizer use be offset by this biological solution. They're not the first ones to have thought of something like this. Pivot Bio is a Bay Area company that is also looking to replace synthetic fertilizer with engineered bacteria. They have a slightly different approach to how they engineer the bacteria, but the end goal is very similar. Now, they do have some data that show that their product, which is called Proven, is able to help plants grow better with less fertilizer. So you kind of offset some of the fertilizer with this Proven product, this bacterial-based product. But in Nebraska field trials, which were released pretty recently, the proven benefits are limited. It looks like adding the kind of engineered bacteria didn't really help the plants grow any better. And so it's unclear exactly what happened, but one common thing with engineered bacteria is that they don't live for very long out in the environment. You can engineer bacteria in the lab to do some pretty amazing things. But as soon as those bacteria are put into a competitive environment where there are, hundreds or thousands of microbes competing with your engineered bacteria for resources, then your engineered bacteria may not survive. It may not be able to continue to do things like fix nitrogen once they're out in the wild, if they're having to continue to compete just to survive. So it would be nice to see in some of these trials measurements of how much bacteria were in the soil at the end of growing seasons, and to see these repeated. These were all done in Nebraska and all with corn. And so it would be nice to see some kind of other data for these types of products, because at least in the Pivot Bio team's trials, they did seem to work. So it's important for us to figure out how all of these biotechnological solutions are working, how well they're working, and what kind of situations they're best for. Okay, one kind of off-the-wall solution is just to engineer the plants to self-fertilize by fixing their own nitrogen. So instead of figuring out how to get them to associate with bacteria more tightly to form root nodules, or how to get the free-living bacteria to fix more nitrogen, maybe just engineer the plant to produce nitrogen itself. Now, that sounds like a good idea, except these nitrogen fixation pathways, the enzymes that fix nitrogen, are very complicated. Here are two examples of gene clusters from bacteria that encode for nitrogen fixing enzymes. And each of these little arrows is a gene, and you can see that there are lots of them involved in this process. And so figuring out which ones you need to move into a plant in order to get the plant to fix nitrogen is not easy. People have taken some first steps at this and have been able to get some nitrogen fixing enzyme components into plant cells and verify that they are functional. They've not yet gotten full nitrogen fixing to happen in plant cells. I think this is actually in yeast, so in eukaryotic cells, but they're on their way, and that's really exciting. So it would be great to just have the plants fix nitrogen, and that could solve a lot of those nitrogen runoff problems too. If the plant is just making what it needs in terms of nitrogen to grow, then you would worry a lot less about there being too much in the field, and then that nitrogen getting into other ecosystems and disrupting them. So kind of cool. All right, so that was your crash course introduction to plant biotechnology and some sort of human nutrition. I want you to think about where the protein in your diet comes from. Can you access information about what the amino acid composition of the foods you really like to eat might look like? And can you tell, are you getting enough of the essential amino acids that your body needs to survive? I encourage you to kind of think closely about your diet and what you might change if you find that you're not getting the right amino acids for optimum performance. Do you view the invention of synthetic fertilizer as good or bad? We talked about some pretty significant benefits and some pretty significant drawbacks to this type of invention. I'd like to know what you think about it. And then what role did semi-dwarf plants play in the Green Revolution or in this massive increase in food production that happened in the mid-1900s? And then which biotechnological method of reducing synthetic fertilizer use are you most excited about? We talked about 3 different ones today. Maybe you're more excited about one than another, or maybe you have your own idea of how we can solve these synthetic fertilizer problems. Okay, see you in the next lecture.